So thank you everyone for joining in on this warm sunny day and taking the time to listen in to what I think will be quite an interesting presentation. So we're going to let the speakers introduce themselves, but I think the topic is really interesting, and I've seen V present previously, so I'm really looking forward to this, and I have been for a while. So take it away, speakers.
So I'm gonna introduce Dr. V. Mohamed. She did her PhD in UDM, studying tauopathies in the field of Alzheimer's disease. Then when she finished, she moved to the neuro to study synaptinopathies in the field of Parkinson's disease. And she was the one who started the brain organoids technology at the neuro. She was exactly the first person that decided to start with this very novel technology. She's a great postdoc. She has very good potential in the research. When she's not in the lab, she's at her bakery because she owns a bakery as well with her husband. And also she loves to do exports outside, the kind of exports that bring your level of adrenaline up with like alpine ski or rock climbing. She's a very active person and a very good scientist and a very good friend as well. So it's your turn.
Thank you, Maria, for the introduction. So hi, everyone, today. First, I'm going to share my screen and hopefully it's going to work. Okay. Right. Do you see well the presentation? Yeah. Great. So the method I will present to you today is about a rapid scale-up in midbrain organoid generation. As you know, studying Parkinson's disease in the laboratory presents many challenges. The main one being the limitation to access the tissue from living affected patients. And so the emergence of the induced pluripotent stem cells, also called iPSCs, allow for the differentiation of neurons and glial cells, for example, in vitro in a dish, from a simple skin or blood samples coming from the patient. However, to reproduce the complexibility of the brain's environment, the researchers are developing more refined iPS-based tool to model better Parkinson pathophysiology and to advance drug discovery. Recently, the development of cerebral organoids is a major technological advance in the stem cell field. And first, what's an organoid? So Madeleine Lancaster in 2013 generate this tissue that contains many features of the brain. And so cerebral organoids are composed of a mix of neurons, astrocytes, and oligodendrocytes, which are self-organized in the space. They also exhibit cerebral function. You can then observe saltatory transmission, node of Ranvier, and synapses. And since 2013, different type of brain organoid have been generated based on the adaptation of the initial protocol published by Lancaster. And now with the modulation of specific inductive factors, we can give rise to multiple types of brain, including, for example, here, midbrain organoids to study Parkinson's disease. And so the differentiated organ cell types can be generated in vitro from iPSCs. We follow basically a series of differentiation steps that mimic early embryonic development with midbrain differentiation factors, and by playing with physical elements such as ultra 96-well plate, ultra-low attachment made, a scaffold of matrigels to provide an extracellular matrix for the tissue to grow, and shakers to provide some motion for long-term culture that will help the tissue to form and to stay alive. So you can basically expect to see the organoids growing from day 1 to day 50 in culture and reach approximately 4 millimeter in diameter. And I used to produce organoids with 96 well plates and maintain them in culture in orbital shaker with 6 well plates stacked on each other. This technique, using the 96 well plate and the shakers and 6 well plates stacked on each other, is now extensively used in labs. But this technique has actually many disadvantages. To generate the tissue first, because we are working with 96 well plate, it requires a lot of pipetting. And also when we have to provide the extracellular scaffold for the tissue to grow, it's like single embedding of the tissue. So again, a lot of pipetting. Then we have to single transfer them into the plate. So again, a lot of handling. Because of the 6 well plate format and the quantity of organoid we used to do, the maintenance for the media is also very heavy. We used to change the media 3 times per week, and I'll let you imagine when we have like several shakers, several incubators, several hundreds of plates, the time that it requires to maintain all of the material. And so for all these reasons, I refined the process of the midbrain organoids generation, and I submitted a manuscript for peer review a few days ago. So the preprint is now available on bioRxiv at the following link that I put here if you want more detail after this talk. And so overall, my goal is to ameliorate the organoid production, making it robust, consistent, and scalable to levels where organoids can be provided to users across the life science sector for research and drug discovery. So in this manuscript, I describe a novel protocol using a microfabricated embryonic bodies device called the EBDisc to generate large batches of homogeneous midbrain across multiple different iPS lines. I derive midbrain from different iPS lines using the classical midbrain partnering factors following previous publication and following previous timeline that I mentioned in my first generation paper. To generate the midbrain from a single EBDisc, the device containing 948 microwells, I start from iPS colonies at 80% of confluency. And the process to generate midbrain from iPS cells took about 8 days. After dissociation of the iPS cells into single cell suspension, I seed the cells onto one EBDisc containing the 948 microwells in order to generate 948 midbrain organoids. And I centrifuge this disc to form one cellular aggregate within each microwell. The subsequent step consists of driving the cells toward a midbrain fate with several media changes containing the differentiation factors of interest in a stationary culture until the HMO, the midbrain, were embedded at day 7 directly onto the disc. So I just put 1.5 ml for that disc of matrigel to provide a scaffold to the organoids and to promote the tissue development subsequently. Finally, on day 8 here, I lift up the disc and flip it into a dish containing the final differentiation media to promote the growth of the tissue. And the matrigel embedded midbrain organoids were then transferred into bioreactors containing the final differentiation media to promote their maturation and to maintain them in culture prior to the collection at defined time points for the experiments. There is something very critical to monitor if you want to make organoids. I actually observed that the condition of the iPS cells is a primary determinant for successfully generating high quality of midbrain. So iPS colonies were maintained daily and passage in order to present with minimal areas of differentiation, because you only want to keep the iPS that are still pluripotent, not to having contamination of cells that will have lost the pluripotency. I use several discs containing the 948 microwells in parallel across different lines and synchronizing confluency to generate big batches with different conditions. And the aggregates in each disc were then transferred into a specific bioreactor and placed on a magnetic stirrer house within a regular cell culture incubator. Each magnetic stirrer can hold up to 9 bioreactors. So this is a video of the setup you could expect then. And this is more information about the disc itself. So the generation process that I describe in this manuscript to scale up the production of midbrain was based on the use of a microfabricated device, the EB disc, that I depict here in the figure in A. So for the production of batches with several hundreds midbrain, I use the EB disc containing 948 microwells. I made them, I designed them in partnership with Nguyen-Vi, which is biotech based in Montreal. And we pre-coat them with an ultra-low attachment coating to prevent the cell to stick to the dish. And this coating is composed of 5% of pluronic acid. And the disc has precise defined dimension, as you can see here on the middle pictures. The volume of each microwell was like 1 millimeter cube, while the microwell diameter here was 900 micrometer. The discs were made from PDMS, which is a flexible material permitting like easy handling of the platform for the midbrain organoid production, while being biologically compatible with the neuronal tissue. So I observed the progressive appearance of the aggregates within the disc from the seeding day D0 to the transfer day D8. So indeed, after 48 hours in the disc, EBs, embryonic bodies formed and present with very smooth edge here. Then they keep going to grow up and they start to present neuroectodermal bud extrusions around day 7. The embedding on day 7 with the matrigel led to a more pronounced bud extrusion on day 8 here, 24 hours after the embedding. And the following day, they are transferred into spinning flask bioreactor to promote the growth of the organoids. So I quantified the area of the EBs and the midbrain organoids. They are kind of derived from 5 distinct iPS line, including here 2 Parkinson's disease line. And I compared the size from day 2 to day 8 within the disc using an in-house cell profiler pipeline that creates color mass here to visually verify the area to be quantified. Our analysis demonstrates that EBs from all lines grew up in size from day 2 to day 8 to attain approximately 500 to 600 micrometer in diameter at day 8. The area of this midbrain generated at day 8 from the disc were quite similar across all lines. And this is what you can expect to see visually from a freshly transferred midbrain. Compare here to more mature one. So this one, it was like very few days after they were transferred. They are very tiny. It's the head of a needle. Well, here you see when they are growing, they are becoming quite big and obvious by eye. Okay. So after 10 days in the spinning flask bioreactor, I count the midbrain organoid from 2 different backgrounds here, and I count respectively 650 midbrains here and 700 and something midbrain here in this bioreactor. So the yield of the material obtained from a 948 microwell EB disc was approximately 65 to 70%. So I note the fusion of some midbrain organoids that account for numbers being lower than our starting number of 948, as well as some midbrain being lodged in the bottom of the spinal impaler. Within each cell line, I also note an homogeneous size for the organoids when I collected them and fixed them at 100 days. To assess the cellular composition of the midbrain, I perform flow cytometry analysis from one control line that I dissociated into single-cell suspension after 50 days in culture. And so the number of cells per midbrain was roughly on average 100,000 cells after dissociation, and the viability around 81% across 2 batches, which was very good. Additionally, to assess the diversity of cell population within the midbrain, I stain cryosection for markers of astrocyte, S100 beta, markers of neurons, MAP2, markers of dopaminergic neuron, TH. And so additionally, to verify the midbrain identity with markers such as TH by cryosection, I also run qRT-PCR to look at midbrain genes, for example, here, NER1, NMX1A, 1B, DDC. And as you can see here, they are present in the midbrain and absent in the parental cell line iPSCs. Conversely, I could detect iPS genes markers such as OCT3, 4, and non-organ iPSCs, but not in the midbrain organoids. Additionally, after 100 days in final differentiation media, the midbrain organoids start to develop and accumulate spontaneous neuromelanin granules. And this observation was previously confirmed to be a feature of functional dopaminergic neuron. Next, to further validate the midbrain identity and the reproducibility of this new method, Maria Lacalle clear and stain 4 6-month-old midbrain from our control line, and she stained for TH, the marker of dopaminergic neuron, and MAP2. And so consistent with previous observation, she was able to confirm the presence of dopaminergic neuron as expected for midbrain organoids. And now I will let you appreciate the beauty of this 3D staining that Maria will reveal to you in the second part of the presentation. She will let you know how she does such amazing imaging of the organoids. Finally, the volumetric quantification of MAP2 NTH staining percentage showed that similar levels of neurons and dopaminergic neurons were present across each of the 4 midbrain organoids tested. And the TH MAP2 ratio show a similar yield of dopaminergic neurons in the 4 midbrain organoids. Taken together, our findings indicate that large numbers of midbrain organoids can be generated with this protocol. The midbrain presents good cell viability, a midbrain identity, and a similar cell type relative to each other. So I don't know how you want to proceed, but maybe we can keep the question for the very end of the presentation. And I will now let Maria Lacalle handle the second part of this talk. Few words about Maria. She made her PhD in Spain. She was in an imaging team, so she has extensive knowledge about microscopy and stainings and imaging in live and in fixed tissue. Then she came from Spain for her postdoc at the MNI to work on mice and diabetes, among many other topics. She is now a research associate in the early drug discovery platform, and she likes vasculature topics, cats, and she grows amazing flowers. She also has magic hands with organoids that she turns completely transparent to better reveal them. So this is the method she will explain to you guys now. So go ahead, Maria.
Thank you. Okay, I'm gonna share now my screen. Okay. Yes. Okay. So now I'm gonna show you how I do these magic things. It's not magic; it's just science. To make the organoids transparent and to do the 3D imaging and how we process those images to get the information that we need for our comparisons in our experiments. So as you already know, in the way we can study the anatomy of brain organoids is either by the macrostructural feature based on the shape or the size of the organoids, or based on the site architecture, where we compare the cell distribution in the tissue or the kind of cell types that start appearing at different time points in the maturation of the organoids. But when we approach this study through the conventional 2D images, on top of being very time consuming to produce so many sections, to cut all the sections in the cryostat and do so many immunofluorescence, on top of that, we also increase the variability between organoids and it makes it difficult for group comparison because we have too much variability due just to the limitation of studying this architecture based on 2D section. For example, what I mean is that if we cut some section at the superficial layer, for example, we see that there is a high content of mature neurons with MAP2 labeled here, and we may find other sections that have very low amount of mature neurons and mainly progenitor neurons in the core. Then if we pull this data together, means that we have very, very large variability. And what happened is that these organoids, for example, at 2 months of maturation, they follow a radial distribution. They are like an apple. So in the outside part, it's full of mature neurons, and then in the inner part is more progenitor. So how to reduce this variability and being able to find differences between groups without the need of cutting many, many sections or using many, many organoids to reduce this variability. So the best option is to image the full organoid and get the entire information from each organoid. This way, the variability is very low. And as you saw before, with only 4 or 5 organoids, you can see it's very, very reproducible. the data, as V shows before in her presentation. So for tissue clearing, I put this picture because when I was just starting to do the tissue clearing, I found this after the rain one evening that was very raining. I found this drop of water on top of my plants in the balcony, and I thought, oh, this is exactly my gold standard. This is how I want my organoids to look when finally I can optimize this method, this protocol. Okay, so what is exactly what we do for clearing the organoids? It has this tissue clearing protocol has two major steps. One of them is to remove the lipids of the membranes, the cell membranes, because they produce a lot of light scattering, meaning that the light that comes from your microscope, for example, is going to be reflected. You lose light in the objective, and then it is what we see as an opaque, the opacity of the biological tissue. So when we remove these lipids, then we maximize the light that can penetrate through the tissue, and that makes it more transparent. So this is an example of what happens if you do not clear your tissue. You are limited to the superficial layer of your samples. You cannot visualize more than maybe 100 microns. But when you remove the lipids, then you can visualize like 600 microns or so. Okay, so what is the second step? As the same way that lipids are producing a lot of light scattering, also the water has some light scattering. So what we do is to remove the water. We dehydrate the sample by using oily or sugary solution. What happens for our eye to distinguish between two different samples, like it's that the light that insists in your sample is gonna bend because it's gonna get stopped, and then it's gonna change the speed and the direction, and this is what our eye perceives as a different material. So, for example, in this glass pipette that is empty, you can see the difference between the oily solution in the jar and the air that is inside of the pipette. However, if we fill this pipette with oil, the same oil, then we no longer see the cannula, the glass pipette inside. So this is basically what we do with the organoids. Then we will incubate them inside of an oily or sugary solution. So we will image them directly in that solution. So there is no transition between the air and the organoid. And this is an example of a mouse brain, for example. That was the first time that was published, this kind of protocol in a Nature. And we are doing exactly the same with the organoids. There are different kind of protocols that can be used to do this. Some of the protocols are using organic solvents. They are very toxic. It's not good to use this kind of protocol. And also they bleed a lot the samples. So you have a lot of quenching of the fluorophore. You can use also water and sugar solution like glycerol with fructose, for example. And then this is to make them transparent, but you don't remove the lipids. So it's a bit transparent, but they're not perfect. There are other protocols that what they do is to use detergents to remove the lipids first, and then they use another solution to remove the water. So it has two steps. This one of them is, for example, cubic. That is the one that we are using here. And there are other kind of protocol also that are water-based protocols, but they have an additional step to protect the protein. That's why it's called tissue transformation protocols, because they build a kind of a scaffold of polyacrylamide, for example, to protect the proteins to be washed during the detergent based steps. Okay, so here is an example of how they detected what were the best cocktail of reagents to remove the lipids for the cubic protocol. They just mixed some mouse brain tissue. They incubated with different concentrations of tritone, and then also they add urea because it's a mixture. And they saw that the transparency of the solution at the end, it was optimal with this combination of tritone and urea. They added on top of that some polyhydric alcohol. This mix of specifically quadrol, tritone, and urea is optimal to clear the sample. Plus, it is very gentle with the fluorescence, so we won't lose the fluorescence in our sample. Actually, we can keep those clear tissue for many weeks, and still we will be able to image them later because the fluorescence will be still there. The other advantage of using this protocol is that it's super cheap, and you just need a transport tube to put it in a shaker at 37 degrees. For 1 or 2 days, the organoid will be clear. And then this is an example of the protocol that I'm following. It was optimized for chicken embryos brains. And we are just reproducing exactly what they did with our organoids. This is, so in 2 or 3 days, we remove the lipids with the reagent 1, water, urea, quadrol, and tritone. Then we do the immunolabeling. This is good because once you remove the lipids, the tissue is gonna get hydrated. It's gonna get a lot of water. It's gonna expand a bit, but that is gonna make it easier for the antibodies to penetrate. Then after that is when you dehydrate with sucrose, and then you incubate with optical clearing tissue. So in the first step, you get some translucent sample, and at the end, you get the transparent sample. This is when you don't have water. For imaging, there are different ways you can image these tissues. Light chain microscopy was designed specifically for 3D imaging. It has a very good resolution. There is very low photobleaching because the light comes only in a sheet. It's a beam that is a flat, and the acquisition is made in parallel with the focal excitation at its step. However, in the case of the organoids, because the cells are not exactly homogeneously distributed, sometimes you will have a lot of cells in one area, less cells in another area. That makes a lot of light inhomogeneity that, well, it can be corrected with some protocol, some algorithm for image processing, but it's not ideal. When you already have a bad image, it's difficult to really get something good out of it. Another complication of this kind of imaging is that because the light comes only from a focal plane is completely sheet, then any kind of particle in the mounting media, or even in our case where we have some neuromelanin, it will create these kind of shadows that in the end you will have an image full of dark lines. And again, it's a complication for the quantification of the volume that you want to quantify later. Some microscope improve that kind of artifact by pivoting the light, so it make a little shadows, so it reduce a bit the dark lines that you get. This is one of these microscope that does that. It can be found at the Bellini. But yeah, taking images of the entire organoid takes very long. The images are very, very heavy, so it's difficult also to handle them later for regarding the analysis. Because the organoids are small, are very small. If they are less than 1 millimeter, they can be used. They can be imaged at the confocal, a regular confocal microscopy. This is an example of two organoids that have been imaged with using the Leica confocal at the neuro. One of them is 2 months old. You can see the radial distribution. It has transcription factor SOX2 for progenitor neurons, and then outside it has the TUTS1 for mature neurons. So it's completely radial at the beginning, and then as it matures, when it is already 6 months, these are actually exactly from the same batch. Organoids taken from the same batch here at 6 months is the one that V already showed in her presentation. You can see that there is the mature neuron has been distributed everywhere. It's there is a higher content of mature neurons at 6 months. But again, if the organoid is bigger than 1 millimeter, then we are limited by the focal distance in the objective in the microscope. So we cannot acquire the entire organoid. And then we can do it at the Opera Phoenix. It's a high content screening system. But okay, so the resolution is very good. It's almost like a confocal. It has the same kind of resolution. This is an example of an organoid 4 months old. I didn't put it here, but it's 4 months. You can see mature neuron with tau, and then TH for the dopaminergic neurons. This system is very fast. What you could acquire in 2 hours or 3 hours in the confocal, here it can be acquired like in 5-6 minutes maximum. And also you can do the acquisition of the 4 different fluorophores. You don't need to do it sequential. You can do it at the same time because it has a multi-pinhole disc that rotates very fast and separates completely every how do you say that, the excitation of the fluorophore. So there is never a crosstalk of the fluorophores. And this is good because you can image the organoids in a 96-well plate. So you could potentially image many different organoids at the same time, different studies that you want to do. But still today we have to optimize this part because sometimes, because it has an autofocus system, it doesn't really recognize some of the organoids. So some of the wells fail and we miss all this information. So we are working on this. We need to really improve this part still, but it's a good option to do images of 2 millimeter or 2.5 millimeter organoids. Okay. And now about the quantification, it's actually very simple. It doesn't really add more work of the kind of analysis that you would do in a 2D section. The thing is, before you could analyze its section by section, as the number of pixels that are positive for your marker. So in this case, now we have, instead of a pixel, we have a voxel because it has the 3D. So it has a third dimension, but it's exactly the same. The concept of how to analyze this, instead of just multiplying the pixel side by the number of pixels that you have positive, you just multiply the voxel side by number of voxels that are positive. This can be done with ImageJ by using voxel counter plugin. Or I also did a MATLAB script where you introduce the voxel side and it will go through all the sections and it will give you the results. So both of the programs are good to use for this kind of purpose. And this is a bit what you do to do the quantification. You just take your sample, to split the 3 channel, and you generate the binary mask. So you just get what is sample, what is not sample. So the pixel value will be only 1 if it is positive, or 0 if it is negative, it is background. So this way it's easy to compute the number of voxels that are positive per voxel side. And this is how I calculated the values for this paper. And now, because the idea is to be able to quantify many samples at the same time, like 100 of organoids, if we ever manage to image them in the Opera Phoenix. So we have developed an application in MATLAB to accelerate the image processing. So here you can see this is nuclei, and this is motor neurons. You just present a threshold to recognize the area. And then it gives you all the voxel side, and you can calculate the ratios between the nuclei volumes or the motor neuron volume. And here you already have the mean, the standard deviation of its experiment. It's a set of organoids. So that's it. The cubic protocol is a cheap, non-toxic, and easy to implement method for organoid tissue clearing. 3D imaging represents a promising tool to better identify pathology-driven alteration in organoid cell architecture, and tissue clearing facilitates the scale-up of imaging and processing of 3D volumes. If you ever want to use any of those techniques, or you want to image your sample in any of those microscopes that I told you about, just contact me. I can give you some advice. Yeah. That's it.
All right, thank you so much for both of our presenters. That was a really great talk, or set of talks. So I guess one thing that I'd like to do is open the chat to anyone. If anyone wants to add questions of their own, you can also just raise your hand or come into the discussion. I have a couple questions of my own, but I'll leave the floor open just a minute so that if you do have a question, you get priority as as a non-host. So we have a question from Alba.
Hi, good afternoon. Thank you for your talk. A question about the imaging with the Opera system. What will be the resolution of that imaging?
So it's high. It's 2000 pixel per. You can. It's not like in the confocal where you can play with the resolution. It comes from the camera that it has, but it's 2000 something per 2000 something exactly, depending on the objective that you use. But for the 10x that I used, it's about that. I don't remember exactly the number, but it's not exactly the one that you can find defined in the confocal, for example.
Okay, thank you. Do you have the 60X?
In the Leica? Sorry, in the
Opera.
yeah, in the Leica for sure, but in the Opera I haven't tried because we don't want to go more than 10. Otherwise, the images are too big. I think that now with the 10x, each image it's already like almost 3 or 4 gigabytes. So it's crazy to analyze those things. So yeah, but if you want to go to a small portion of your organoid, you can still use the 20x, for example. It's good enough to have a better resolution. Yeah.
Okay. Thank you very much.
Okay.
And Morgan?
Thank you so much for the great talks. They were very informative and your work is really exciting, and congrats on the recent submission too. My question was about the percentage of other cell types in the organoids. Like, are there, how much astrocytes and microglia are in them, if any? I was just curious.
Yeah, so actually in the first paper where we generate the midbrain organoid, we did a single cell RNA sequencing to better characterize the diversity of the cell population. And I only mention astrocyte or oligodendrocyte, but there are actually plenty of other populations, such as radial glia, apparently astrocyte and astrocyte-like other type of glial cells as well. But what can I tell you is it's around 60% of neurons
Nice.
or progenitors of neurons, depending on the age where you are looking at them. And yeah, roughly 40% of glial cells. Yeah. But this ratio, it changed over time during the maturation process. And also it might change depending on the genetic background. Because, for example, I'm working with Parkinson's disease line. And I can clearly see that midbrain organoids derived from Parkinson's disease patients. Sometimes they do have like a decrease of the dopaminergic neuron numbers or an increase in the astrocytes because there is a reactive profile and an inflammatory-like profile. So this is something to keep in mind depending on what you are looking for exactly.
It's really interesting though that in the disease model you see a lot of proliferation of the glial cells. My lab studies Alzheimer's, so we study neurons and microglia and things like this. So that's why I was curious.
Yeah,
really cool.
Thank you.
Thanks.
So I was also curious. I don't actually work with neurons, so this question might just be already assumed by the people in your literature, but would there be any benefits from making iPSCs from cell types that are phenotypically more similar to a neuron? Because I would assume you're taking from fibroblasts or something like that.
Yeah, so basically when you do iPS cells, you reprogram the cells and it can give rise to any type of cell of the body. It could be a neuron, it could be a cardiocyte, a myocyte, whatever you want. And so we just put inductive clues to drive them toward a neuronal fate. So yeah, because we study, for example, for Parkinson's disease, this is the midbrain area which is affected. So we differentiate them into neurons and glial cells and other for the organoids. But for example, for Parkinson's disease, there are plenty laboratories worldwide working on other aspects. For example, the gut-brain link for Parkinson. So it's very nice to be able, from the iPS line, from a specific patient with a genetic defect in the background, for example, to differentiate them into midbrain organoids and also, gut organoid, for example, and make a link and look at other type of mechanisms involved in the disease. So Yes, we are mostly midbrain organoid orientated now in our lab, but we don't exclude at all to generate in future gut organoid and other type of organoids that may be involved in the disease.
Okay. And just before we go on to the next question, would it be possible to generate kind of growth factor gradients using what you're doing? I mean, I know you're doing, everything's done in rotation. Are other labs kind of doing it in more of a solid form so that you can get those kind of more complex structures?
Absolutely. So in development, you have gradients of inductive signals and growth factor to have a spatial organization of your brain, for example. So there are actually labs working with beads where they put some growth factor, and they, with a magnetic system, they stick basically the beads to one side of the aggregates at the very beginning to increase the concentration of the growth factor at these sites compared to the other sides of the aggregates. And so they try to make structures that are more dorsal on the side and more ventral on the other one. So yeah, this is a works ongoing. It's super exciting. It's very pioneer works for now, but I believe that in a couple of years we will be able to maybe make better organization of a better spatial organization of the brain organoids.
Okay. Isan, you had a question?
Hi, yeah. So thank you for the talk. It's such a cool topic. I was wondering, do you find that there's a lot of variability in between organoids and in between batches? And did you find any ways to kind of mitigate some of these parameters?
Yeah, so first, it depends on the type of organoid you are doing. If you do cerebral organoid and you use the initial protocol published by Lancaster in 2013, because there is no inductive signal in that protocol, the cells going to differentiate by themselves. And of course, you're going to end up with very variable tissue. The most recent protocol using inductive signals will give you a better quality of batch at the end, because you're going to drive them at day 2, day 4, day 7. You're going to put like at very precise time point a certain amount of your inductive signals. So that will definitely help for batches that are more homogeneous. What I can tell you from my results so far is, even though they are different from one to another one, with about 12 organoids for each set of experiments, I was able to see clear tendency in my results and across batches. So this is something to keep in consideration, that to work with at least 12 N and to be able to see it across different batches, it's something really important for your data when you want to look at phenotypes or mechanisms for your subject of interest. And now with the method that I presented today with the disc, because we constrain them in size with the disc, they are smaller than the initial protocol. It means that the growth factor that penetrates the tissue the penetration is much more homogeneous. And I believe that's the reason why when Maria did the quantification of the cell population over 4 different midbrain, she saw something quite homogeneous. So that was actually very encouraging in terms of improvement of our initial protocol that we published a few years ago. So yeah, I hope it answers your questions.
Yeah. Thank you so much.
You're welcome.
So Morgan had a question, and then I have one more, and then we'll kind of be running a little over time. But if there are any more questions, you can just put them in the chat, and we'll try to answer after.
Okay, thanks. My next question was about the epigenetics, and if you're able to preserve the epigenetics of the iPSCs to the differentiated cells. I've never worked with iPSCs, so I don't know if this question is any good, but I was just curious about that part of it.
Who do you want to answer, Maria?
Don't know about the epigenetic factor in these organoids. Yeah, no idea.
So I think this is the very hot topic in the iPS fields, because when you do reprogrammation of differentiated cells into iPS cells, you lose the epigenetics, right? You reprogram everything. So there's a reason why also we don't work with sporadic cases, sporadic lines. You know, we work with iPS lines that carry genetic defects known to induce the pathology. That's the reason also why people are now making what we call transdifferentiation. So they don't go to the iPS stage, they don't do reprogrammation, they go from a fibroblast to neuron directly. So this is another method to preserve the epigenetics. And there are people working on sporadic disease line doing that method. It has advantages and disadvantages. The advantages is to preserve the epigenetics. The disadvantages is you cannot do proper isogenic controls because you are not capable of doing CRISPR-Cas9 on transdifferentiated cells. And also the yields might be very low, for example, to get dopaminergic neurons. So it's 5% that you're gonna end up. So it really depends on your approach and what you want to do with your material. So yeah.
Thank you. It's really interesting.
Thank you.
And so one final question for Maria before we end it. I was kind of curious what you think are, so I mean, I know that we're kind of coming up on a limit, a physical limit on image resolution for confocal imaging, or at least that's, I've heard that. I'm not exactly all that in on the field. So do you think they're going to be the biggest advancements in imaging for small organs or organoids? Are they going to come in the field of staining, or do you think there are going to be advancements in imaging that will really bring us forward?
Yeah, no, it's going to come from the optical part. Yeah, it's just improving the speed of the light sheet microscopes. I think confocal is not ideal. I think light sheet microscopy is the best right now, but we need a microscope that go faster and that are also able to compress the size of the images and these kind of things, but still people are working on it. But yeah.
Yeah, and I mean, do you have any size limitations for when you're trying to do kind of a cubic clear?
The size, yeah, so not really. It just take a bit longer if they are bigger than 2 millimeters, let's say. Instead of 2 days, maybe it can take 4 days, and you should increase the speed of the shaker. For example, instead of being 80 revolution per minute, you can increase to 100 and 120, for example, to still be gentle with the tissue, but move them a bit faster. Then it's fine. Yeah.
And you've never had issues with antibodies not getting all the way through, or that kind of?
Yeah, you need more time as well. Instead of using 2 or 3 days, you will keep them for 1 week, and you have to refresh, adding extra antibodies day 3, for example. So you do day 1, and then at day 3 or 4, you add a bit more antibodies, and you keep it for 1 week. Yeah, it's just about time.
Yeah.
Yeah.
All right. Well, thank you so much for your presentation. I really enjoyed it. And I think the others did too. I got a couple direct messages. And so I'll also be posting this later on the EMGSS YouTube. So if you ever want to share it with anyone, or if anyone missed something in the presentations, we can always go back to that. So yeah, thank you so much for your wonderful presentation.
Thank you for the invitation.
Yes.
Bye-bye.
Bye.
Thank you.